mutations that cause genetic disease, such as sickle cell anemia, can be identified using a DNA test called allele-specific oligonucleotide hybridization. In this procedure, a short DNA fragment, the oligonucleotide, also called the probe, is created to match the normal version of the gene, and another oligonucleotide is created to match the sickle allele. The normal and sickle probes differ by a single nucleotide. In this family, a mother, father, child, and fetus are being tested for the sickle cell allele. The sickle cell mutation occurs in the beta-globin gene, which codes for a subunit of the hemoglobin protein in red blood cells. Each person has 2 copies of the beta-globin gene. The normal allele is usually designated A, and the sickle allele is designated S. Both copies of the gene from an individual are amplified together by PCR and then dotted onto filters for testing. The DNA on the filters is denatured to make it single-stranded. The probes are either fluorescently or radioactively tagged to be able to visualize them. The probes are added to the filters to allow them to hybridize to complementary sequences in the spots of DNA, and after incubation the excess is washed off. The probes are very specific. Each hybridizes to just one type of beta-globin allele to make a perfect hybridization match. In this test, the red color indicates that the probe hybridized to DNA on the filter. The blue color indicates lack of hybridization. In this test, the mother, father, and fetus all show DNA hybridizations with both probes, so they are heterozygous, AS. They are carriers of the sickle cell allele. The child, in contrast, has DNA that only hybridizes with a probe for the normal allele. The child is therefore homozygous for the normal allele, AA. Another way that the sickle cell allele has been identified is by using enzymes that cleave DNA. In sickle cell anemia, the sickle cell allele has a mutation that converts an adenine base to a thymine base. Whereas the normal sequence is digested by the restriction enzyme MST2, this enzyme cannot recognize the sequence in the sickle cell allele and therefore does not cut it. In this test, you use PCR to amplify the DNA region that includes the mutation site. If you were to digest both samples of DNA with MST2, the normal allele would produce 2 fragments, and the sickle allele would produce just 1. In performing this test, you would digest DNA from the test sample of interest with MST2, as well as digest DNA from a normal beta-globin sample and a sickle cell sample. The digested DNA is then loaded onto an agarose gel for electrophoresis. The DNA fragments are negatively charged and migrate toward the positive electrode in an electric field, with the smaller fragments moving faster than the larger fragments. From the banding pattern on the gel, what is the genotype of the person from which the test sample was taken? The individual has both alleles of the beta-globin gene and is therefore heterozygous, AS.